I'm Meetali Singh. I'm an assistant professor at the Department of Developmental Biology and Genetics at the Indian Institute of Science. I do work on epigenetics, but I would be frank to admit I'm new to the aging field. So I'm hoping to learn a lot today from the discussion we are going to have. So we are going to have a very diverse audience as per our registration record. So we have students from basic sciences, we have students studying medicine, we have a lot of tech entrepreneurs, healthcare entrepreneurs. So we do have a very mixed, and we also have non-specialists is what I understood. We have members of the general public. So the idea would be that each of these different groups can have a valuable takeaway after this session. And to cater to this diverse audience, we also have very diverse panelists. So we have three panelists with us today from academia, from a genomic entrepreneur, Sudha, and we have also a healthcare entrepreneur. So without further ado, let me first introduce our panelists, and then what I will do is I'll start taking some of the audience questions which we had already received during the registration process. And from there on, we'll go on the discussion, and if the time permits at the end, we'll take up some live questions. So before I introduce our panelists today, a big round of applause for Sagarika, who has managed to put together this webinar with Nikhil's help. So I see Sagarika's video is on. Nikhil, if you can also turn on your video so people know who I'm talking about. And yeah, so Nikhil and Sagarika.
Nice.
So big shout out to both of you. So
Thank
now
you so
moving
much,
on to
Meetali.
our
Nice of you.
welcome. So now moving on to our panelists, we have with us Dr. Germano Cecere. He is a research director at Institut Pasteur, Paris, where he leads the team on mechanism of epigenetic inheritance. Germano is recipient of both ERC Startup and Consolidator Grant and Pasteur Valerie Rado Prize. And Germano's research focuses on the role of small RNA in epigenetic inheritance, and he uses this very interesting model system of C. elegans, which is a roundworm. I'll let Germano talk more about it. And a little bit fun fact, apart from science, in free time, Germano likes to play drums, and he has a band called Los Chacharitos. And very interestingly, he also has an IMDb profile, which I always find very cool as a filmmaker credit. So all of you can probably check it out after today's webinar. And Germano was my postdoc mentor and a dear friend. So very happy to have you here with us, Germano. Thank you for joining. So yeah. Next, we have Dr. Sudha Rao, who is co-founder of Genotypic Technologies and has a vast experience over 2 decades bridging academia and industry. So Sudha has her training as a researcher with PhD from Madurai Kamraj University in molecular biology. And then she went on for her postdoctoral research at Yale Medical School, New York. So in Indian scenario, I think Sudha has been instrumental in establish state-of-the-art genomic facility via her initiative at Genotypic. And this is very, what I would say is very foundational for the molecular diagnostics through her leadership at Genotypic and her new venture at Dhitiomic Technologies, with her work impacting the personalized healthcare and genomic diagnostics in India. So welcome to today's panel, Sudha. We are looking forward to your insights on epigenetics and aging.
Thank you, Lee.
Welcome. And last but not the least, we have Ms. Laxmi Sri Pandrala. She's a nutritionist with specialization in sports nutrition. And Laxmi is founder and CEO of Lami.fit, which is a health-based platform. And when I went over the details, I found it very fascinating that Laxmi is using genomics and epigenomic markers along with the usual blood and body-based body analysis to predict health risk, customize lifestyle alterations to prevent any adverse health impacts and improve the longevity and the health span. And just to mention that Lami.fit has been also the recipient of Women-Led Innovation at the India MedTech Award in 2023. So welcome, Laxmi. Your field is very new to me, so I'm looking forward to learning a lot. So
Thanks.
Thank you. So what I would propose is that before we dive deep into the questions, maybe each of the three panelists, you can talk about your work and research, maybe in 2 to 3 minutes, so that our audience know you better. So maybe I can request Laxmi to go first.
Okay, sure. Thank you so much, ma'am. Really happy to be part of this. I should definitely thank Sagarika and you also, and to give me this opportunity to definitely meet Sudha ma'am and Germano also. So just to give a brief introduction about myself, I'm the founder and CEO for Laxmi Health. So we are a precision health tech company, I would say. So what we do is we collate all the health data of an individual, like it could be their epigenetic markers, it could be the genomic markers, and overlap them with the body metrics and the pathology reports and create that digital health biotwin model. Because India as such, we know we are very much into the reactive till now, into it, right? When it comes to the healthcare, we want to change that perception to being proactive about it so that they have the better control about their own health. So we want them to have that much better health span, not just a lifespan. So we want to bridge that gap between the long lifespan and also the health span. So that's in a nutshell about Laxmi. So we are currently associated with quite a lot of sports organization and wellness sector. In both areas, we are, I would say, definitely known for our expertise. So
that's really interesting. Lakshmi, thanks for giving us this concise overview of your work. Next, may I request Sudha or you to tell us a little bit about your current venture?
Thanks, Meetali. Pleasure to be part of this forum and panelists, especially meeting Laxmi and Germano, and also to hear interesting questions from the audience, because that's what keeps us thinking. My background is genomics, and mostly been in developing technologies for biological research. And so we've been life form agnostic, as in we look at everything that has DNA and RNA, right, from bacteria to humans. But specifically with dithiomics, that is in genotypic. And at dithiomics, my interest was to translate technologies for rare disease diagnostics. It was started in 2014. Essentially being able to use these technologies for detecting mutations that cause rare diseases, genetic disorders. And over the years, I'm sure many of you and in the audience, people have known of these tests becoming a lot more common than they used to be. Simple things like a diagnosis for a rare disease, which used to take anywhere between 5 to 7 years, is now like a matter of weeks. But as Laxmi pointed out, I think we've done that. We've achieved major traction there. And now there is a lot of interest in can our genetics tell us about what quality of life we will be bestowed with, right? So it's not just genes, but we also know that epigenetics plays a significant role in healthy living, right? So it's not just about long lifespans, but the quality of life as well. So that's interestingly an area we will be soon embarking into. So with our knowledge base that we have from actually looking at people who have been afflicted with disease, now translating what we know as diagnostic into something that's predictive is what our next phase of journey will be in dithiomics. So this is a conversation that I'll be very interested to have with colleagues here and the audience. But thanks for having me over, Meetali, on this.
Thank you so much, Sudha. We are actually looking forward to your insights based on your vast experience in the field. Now, may I request Germano to tell us a little bit about your work on epigenetics and inheritance of epigenetics?
Yes. Hello to everyone. Thank you, Meetali, for the invitation, and all of you for this discussion. So I'm based in Paris at the Institut Pasteur, where I led my laboratory of mechanism of epigenetic inheritance, in fact. And so the main objective of the lab is to investigate, epigenetic in the context of heritability, meaning, what we inherit from the past, from our ancestors, in terms of non-DNA inheritance. In addition to our gene, what other molecules can impact our life, in the next generations. And we are using this, so we are working on fundamental principle, in fact, like really trying to understand the mechanism behind this new type of inheritance. And we use this very special organism. It's called Caenorhabditis elegans. It's a nematode, which has been actually instrumental for the aging field. It was the first organism where they finally discovered that there is a genetic pathway that can regulate, in fact, the lifespan of an organism. And in our field, it's been instrumental for studying epigenetic inheritance, because one important feature of the worms is that they are all genetically identical. It's hermaphrodite, so they can self-fertilize. And so we have really genetically identical population. And so you might think they are all identical, but they are not. And so we are studying why they are not identical. If they inherit something, what type of other non-genetic inheritance they have. So this is pretty much what is our topic.
Thank you, Germano. I think you put it in a very simplistic manner for our audience to understand. So now what I will do is, from the questions which we already have, I have taken the liberty of clubbing the questions which were on similar themes, and I'll start posing these questions to our panelists, and from there we can continue on the discussion. And if the time permits, we'll take up some live questions at the end. So you, all the audience, please feel free to type your questions in the chat box, or at the end of the webinar, you can go live and ask your questions yourself. So one very important question, and trust me, it's not my question, it's the questions from audience, few similar questions. What is epigenetics? Because we do have this very fancy title of epigenetics in everyday life. Germano, you did, all of you actually, all three of you did point towards what epigenetics is. But I think it would be good to define what is epigenetics so that all of us and all our audience are on the same page. So may I request you, Germano, to give us the classic definition of epigenetics?
Well, in reality, I mean, to be honest, if we have to be honest with ourselves, we still struggle to find one common definition that can group, what we mean for epigenetics. So in general, what I consider epigenetic is any type of changes that do not intervene into modifying the DNA sequences of our genome that indeed can have an impact on how our genome can, for example, be expressed or repressed and stuff like that. So there is a concept of gene regulation that is known even in non-genetic fields. Now, the feature is that the mechanism that we define as epigenetics, they might maintain this memory of activation of repression across cell division. So there is this concept of heritability that I'm also interested. And this can happen across cell division. So how certain cells might maintain certain gene expression program without changing the DNA sequences, or even what I'm more interested in the maintenance of certain things after, fertilization. And this is more like what I mean is, like what can even be transmitted from one generation to another. So yeah, I would say heritable changes that do not intervene into the DNA sequences that can have some impact.
Okay, I think this was very important because that helps us distinguish between the regular gene expression regulation we talk about and epigenetics, because it has a heritable component. So I have some of these questions, so I am going to read from my cheat sheet from all the questions I have from the audience. So as we begin to unravel the complexities of aging, research has been increasingly pointing to the role of epigenetic in shaping our health outcomes over the time. So given the growing interest in this research area, our next question explores the intersection of epigenetics, health, and aging. So the question which we have from the audience is, how do epigenetic changes impact health and thus aging? And what are these epigenetic changes we are talking about? What are these mechanisms which are impacting our health in aging? So I would like to have opinion of all three panelists on this particular question. May I request Sudha for you to go first and let us know your thoughts on this?
Yeah. thanks, Germano, for laying the ground on what epigenetics is. I think it's also, I think perceptions are that genetics is something that you inherit from your parents, and that's what defines all of what and who you are. Subsequent understandings or research shows that there's one part that you inherit as your DNA sequence, but there's also other things that modify it or regulate it in a way that it can change. So that's why you may not look exactly like your parents, or there are combinations, there are changes that happen. And these could also be influenced by the exposure to environmental factors, right? So for example, I think especially in the context of aging, things like stress, exposure to carcinogens, or, different kinds of UV, many things contribute to how your DNA responds to those influences. And they could essentially then determine how your tissues or cells respond and how they are able to function, right? In that context, I think we look at multiple levels of epigenetic changes. These could be methylation, either of the DNA, or histone, acetylation, methylation. There are quite a number of different changes that can happen on the histones, which essentially change what is available for transcription, or how it is sort of coiled, and it regulates it in that way. Or there could be changes in your mitochondrial genomes. They're the large factories that generate energy for us, and changes in mitochondria could influence the aging status of an individual. And there are other molecules, like, for example, as Germano is working on small RNA modulators, small RNA, then there's long non-coding RNA. Quite a number of those, which again change the information that DNA has in an individual at a generational level, or even can be carried on to the next level. So there are quite a number of those, and I'm sure Laxmi can talk about how she's using those markers for predicting aging for individuals. But I think what has influenced our better understanding of this domain is also how technologies have evolved for us to be able to interrogate and understand all of these mechanisms, especially genomics. We've, I think, for the last 15 years, even with Indian scientists, worked on, I think one of the earliest studies was Dr. Vidita Vaidya's, where she was working on the influence of stress in mothers on what happens to the children as adults. I think these studies have been done, especially during wars in countries. There's a recent study on the influence of pregnancy during COVID, what happened to children. So all of these, I think, have been aided by access to technologies, which can very cost effectively and accurately measure these differences and changes across different molecular types and modifications. So exciting times, and I'm sure we're already on the path to a better learning on how these influence aging. And as you see, it's already getting translated into products that one can use and ask, how old am I, and what do I need to do to stay young beyond Brian Johnson?
Absolutely. Yeah, I think, Sudha, you actually picked up the pulse of audience questions. And I think, as you have indicated, we'll come back to these questions on maternal stresses impacting the next generation. We do have some interesting questions from the audience. So that's really amazing that we are right on that track. Laxmi, may I ask you to put in your inputs on how do you think epigenetic changes impact health? Because you are working with people and you're measuring their epigenetic profiles.
Yes, right. So we could actually, yes, as you know, Sudha ma'am has said that people do think that genetics is only whatever we are born with, right? But epigenetics, definitely, we can see like a lot of information is now available. We can actually see how it is impacting the aging, especially. And that is what people always relate with, that, okay, the age is progressing. So yes, I need to control. But there are so many other things which are affected because of this methylation and epigenetics. Like the stress is also a factor, the immunity levels going back down, how the body repairs is not happening, right? So many things are definitely affected because of this epigenetics. Again, the underlying cause being the stress and the sleep or the diet, the nutrition, so many factors. So at least now I'm happy to be really frank to say at this point, all this information, it is not only just really, confined to the lab or to the research papers. It is at least being able to, we are in a condition, we are in a stage where we have that one more tool, I can say, where we can use it in our day-to-day practice to help those clients, to give those personalized interventions, right? We can also actually have those measurable and trackable changes, which when we are trying to modify, and we can see those changes happening, how the gene, how the reverse of the aging we can actually do. So I should say that that's definitely helping us in that way. So we can see a lot of changes because of the stress, and not just because of, as ma'am was mentioning, people do think that stress, it is only the physical stress or it's only the emotional stress, but eating a wrong food, which can lead to that inflammation, ultimately leading to the cortisol. These are all aggravating that aging, right? So these are also small, tiny markers which we overlook, but they again pose a very maximum risk in terms of the epigenetics.
Okay, I think very nicely put, Laxmi. So what I would like for Germano, can you summarize some of the, so we have, of course, Sudha and Laxmi both have indicated some of these epigenetic mechanisms which regulate these various processes. But if you could, for our audience, which also include a lot of PhD students who are studying various fields in aging and epigenetics, if you could summarize what are the various mechanisms you're talking about in this case, how epigenetics is regulating these aging processes?
Mm-hmm. Yeah, I mean, has been mentioned already by Sudha. There are, what we know so far, like the three major mechanisms are either DNA methylation. This is a mechanism that modify the DNA sequence, but not the sequence per se, but adding a chemical modification on it. Usually DNA methylation is a mechanism of repression. So this is a main important mechanism to repress something in the genome that is called repetitive DNA. Those are transposable elements that can amplify in our genome. Some of them, they have the potential to transcribe themselves, coding for an enzyme that can cut and paste this sequence somewhere else in the genome. Of course, this can be extremely detrimental consequences because they can land into a very important gene. Nonetheless, during evolution, those elements have been fundamental to shape our genome. For example, a lot of promoters for genes that are needed for the organism are composed by those repetitive elements as well. And so there is a balance here. And so mechanisms such as DNA methylation helps to maintain elements that we don't want to jump and get crazy to be in place and be silent. And so this is one fundamental mechanism that in aging also has a huge impact because what they have observed, in fact, is that during the aging process, there is a global hypomethylation that occurs in our cells. So we are born with fully methylated genome with all these repetitive elements, and during the aging, many cells in our body can have a global loss of this methylation, which means that those elements might start to be reactivated with consequences that Laxmi also mentioned about, inducing stresses and so on. So beside DNA methylation, there are also other mechanisms that are in place. And the second one I would say is the histones. So the histones are the basic component where our genome is wrapped around it. Those are 4 proteins that assemble into an octamer. And so this histone now is like almost 50 years of research. They've shown that the tails of these proteins can be heavily modified in specific location. There have been hundreds of chemical modification. The most known are methylation of the histone, acetylation of the histones in different position. And each of those modifications can have different impact. There are modifications that help, like DNA methylation, to repress, for example, repetitive elements and other genomic loci, but also modification that might maintain certain activation of transcriptional program. Again, in the context of aging, the most prominent feature are repressive marks that maybe are being progressively lost, but at the same time, also acetylation has an important role. And in fact, one of the pioneering factor that have been discovered from the yeast is these sirtuin proteins, that is a histone deacetylases that play a major role in longevity. And so there is a combination of global loss of certain mark or removal of certain mark. Acetylation is a mark for activation, for example, that might correlate with the aging process. And then the third one is what I'm more familiar with are RNAs in general. It can be long RNA, short RNA, small RNAs. Those can have a transcriptional regulatory roles. So by sequence-specific manner, they can also interact with all these mechanisms. For example, small RNAs can drive in a sequence-specific manner DNA methylation, for example, or certain histone modification. So there is also a huge, intersection of all these mechanisms that can play a role. These are the 3 fundamental ones, but we are discovering more and more other things. We are lipids that can have some signaling properties, other type of proteins, prions, and so on. They can also have an epigenetic impact on our genome. But I would say, the 3 major ones are DNA methylation, histone modifications, and non-coding RNAs.
Thank you so much, Germano. I think very well put. So we can all agree that our daily life choices and the food we eat, how we manage the stress can have a profound impact on our health. So some of the few questions which we have from our audience are very overlapping with the discussion which we just had. But the questions from audience like, does the quality of the food we are eating can impact our health? Right, because now the vegetables, I think there's one question, if I read verbatim, vegetables, because there are so much of pesticide now. So are the quality of those vegetables going to impact how much we are sleeping, how much that is going to impact our epigenetics and the aging? Of course, then we have questions on stress. There is a new trend of having supplements, multivitamins, protein shakes. How can that impact our health, epigenetics, and then in the process, aging? So I think maybe Laxmi, you could start with your viewpoint, and then other panelists can pitch in there.
So definitely, I'm so glad that I got this question, because this is what we deal in, day out, right? People come to us asking for, do I buy the organic vegetables? Is it really going to affect us, our health in any way? How do I do this? So I would have to say definitely the quality of the vegetables does, right? It definitely does impact the aging, because all the vegetables, the fruits or the vegetables, we are looking at the quality, the content of the phytochemicals in it, right? And all these fiber-rich fruits and the vegetables, they in fact kind of activate the production of these longevity enzymes, like for example, FRN2, right? I'm not talking, NFR2. So then when the quality of the vegetable is definitely not taken into consideration, we can see that is not being activated, and it is not optimal performance your body can give you even after consuming a huge amount of those food. And coming to the next question, supplements. As in my practice, at least of late, yes, maybe around 10 years ago, I would say that we would definitely go for that, dumping that multivitamin left and right. But because of so much of the information available, with the genetics being there, with the genetic information, with the epigenetic information, we have we could now actually come down and narrow down and filter to based on the requirements of an individual. For example, I would rather suggest the person what exactly the vitamin the person is not able to absorb because of their genetic makeup, or because then I would rather suggest them certain vitamins and nutrients which will help them slow down that aging process. Like for example, I can suggest them anytime methyl folate, because that is definitely going to help and reduce that aging, slow down the process. So that would be my take on it. And the supplements, whether good or bad, that is not a question. Whatever taken, whether it is a good, taken in high quantity, definitely is not going to help. It has to be definitely monitored. It needs to be definitely a requirement for that person, whether it comes to that whey protein or whether it comes to that multivitamin, whether it is a vitamin D or a C or anything. We need to understand how much dosage is required for you, and then actually personalize it for that particular person, rather than thinking that yes, this is the best of the vitamin, let me take it, right? So that is my perspective, and that's what we have been practicing, and we could actually see the results, much more faster results compared to where we have given the supplements where the person would not actually, might not be much more beneficial. So we are able to track those changes once we have put the person on these kind of personalized interventions.
Okay, so I think that's very well put. So if I have to just summarize, supplements are good when they are personalized and in balanced dose.
Yes.
Okay, so that sounds good. Germano, would you have some opinion on the quality of diet and stress and how it impacts the epigenetics? Yes.
Yeah, in fact, I would add to what has been said, another very important aspect that is coming out in these recent years, which is the microbiota and the impact of our own set of microbes, either on our life, or there are even recent evidences that can even have an impact across generation. And this is relied on the nutritional aspect, because many of these metabolites and chemical modification that we need, we acquire from the bacteria. And so depending on the composition that we have, each individual has their own microbiome. This can also influence the metabolism of our cells. And what is very interesting in terms of epigenetic is that there is a direct link between all the metabolic pathway and epigenetics. For instance, one very well studied cases is the methylation, DNA methylation, or even histone methylation. The methyl group is coming from the SAM enzyme, the adenosine methylene, and this cycle, it's directly influenced by microbes, by vitamins, and so there is a strong connection on the availability of these nutrients and how they can impact the availability of the substrate that have been used to modify the epigenome. And so this might be one of the direct link with the nutrition, for example. And of course, we have cases of undernutrition. And so like we have strong evidence that come from the early studies in C. elegans, which linked aging with insulin signaling, which is a common pathway conserved up to human, where has been developed the calorie restriction diet, for example. And so, reducing the intake will activate this pathway through the insulin signaling receptor that activates certain type of genes that favors the increase in lifespan of individuals.
Very well said, and I think I also agree with you, or both of you, that diet really have a profound impact. And I like to give this example in my molecular biology course of diet, and how honeybee, queen bee, worker bees are like their path is chosen just based on the diet of royal jelly. Sudha, why don't you go ahead? And I saw that you wanted to.
I just had, this is more for an open discussion, Germano,
yes.
Laxmi, comment on this. So food, I think, is sort of universal, right? And in the recent decades with globalization, I think the localization, which is essentially food that was found suitable based on what can grow in the local environment and to the seasons and the climate has changed, right? So, I mean, pasta and pizza are regular food here in Bangalore. And I wonder if our body's epigenetic marks, which we have inherited, will be not so fine-tuned to these changes in a very short span of time. And could they influence the status of health? Because we do see it's almost a pandemic, right? So there are metabolic disorders that have either due to diagnosis or the prevalence itself has increased. So I wonder, Germano, if you think there could be some kind of contribution there in terms of conflict of changes in food habits with epigenetic inheritance.
Well, absolutely, you're absolutely right. I mean, the epigenetics, in fact, as a concept, is memorized an habit of a gene expression program that can be an habit of, you know, if for certain number of generations, certain individual have been used to certain type of food and intake, so we have our epigenome has responded to adapt to those changes. And so when we dramatically change our diet, this can certainly have an impact on this memory. And then I think you also add an important concept of the heritability. In fact, in terms of metabolic disorder, there is a large fraction of non-genetic components that we still have to investigate of how we might develop certain metabolic diseases. And it might not be directly linked to the food. For example, I might be under the influence of the habit of my parents. So even if I change my diet and I now eat a very healthy food, I'm still might be susceptible to develop certain metabolic disorder for some heritability that come from the previous generation. This has been studied very well in mice. Again, there is a contribution of microbiome in this perspective. What we are missing is to understand what are the drivers, the molecules that are really inherited. We know that there is an impact. We don't know the specific mechanism and genes that leads to certain type of metabolic disorder. But there is certainly a very important connection, as you said.
Thank you, Sudha. I think that was, again, a very right-on question, and thanks for bringing this point for the discussion. So while we are at this topic of heritability, so we did have these questions from the audience about the role of epigenetics and inheritance, and Germano, you already talked a little bit about that. So I know there are a lot of studies, there are a lot of studies in model organisms where different research groups have looked at the heritability, not only intergenerational, but also transgenerational heritability. But there are also a lot of studies on human cohorts. Like the famous is the Dutch cohort during the World War II, where in the grandchildren of the people who suffered during the famine and the drought, they have an impact on the lifespan and the health span. So maybe this is something which I would like you to comment about. So maybe I can request again Germano to start, and then Sudha and Laxmi, you can pitch in your viewpoints on that.
Mm-hmm. Well, I would say that most of what we know so far is some intergenerational effect. In fact, we need to distinguish between inter and transgenerational.
Very important.
This is very important because what we are inheriting, in fact, is information coming from the gamete. That can be the oocyte and the sperm. And so those germ cells are already, formed in an individual. And so when you have pregnancy, for example, the same, the person we are holding can be already, directly influenced by the chemicals that we eat, for example. And therefore, for 2 generations, we cannot talk about transgenerational because in 2 generations that was already directly exposed. So when we talk about heritability, it's something that there was a signaling in some generation, and then without this signaling, the message continue. to be inherited, okay? And so that's the concept of transgenerational. So in case of pregnancy, we should always look at effects that are post 2 generation after. This is an important definition. And right now, most of the effects that we observe in mammals, for example, do not persist for long. There are several explanations of why. And instead, in plants, for example, and in C. elegans, in the nematode, we do have phenomena that can last for multiple generations. Sometimes we observe things that can last even for 10 generations, okay? So this might rely on the fundamental mechanism that we have to maintain this memory. So everything should work with mechanism that after the initial trigger should continue to deposit certain histomodification, if that might be one mechanism, or in the case of the worm, we have a small RNA system that can copy these small RNAs for long time and amplifying the signal for long enough. So we are still at, at the basis of this process. So we still don't know whether in mammals, for example, there might be some longer effects or not. But in the case of your example that you mentioned, the femine, sometimes we might have effects that are a direct metabolite or a direct signaling that is inherent directly in the embryo. So in that case, we do not have to implicate the canonical epigenetic mechanism I mentioned. There is nothing about it. There is simply some chemicals that can be a metabolites and lipids that have been inherited in the embryo, and these can indeed instruct again the next generation in the very early phases. And in fact, the early embryogenesis, it's a very important time window to for the heritability of certain diseases, where we can have, consequences later in life. Okay, and this might explain, in fact, why individuals that eat the same food might develop, different disease outcome, because maybe there was an event during the early pregnancy and embryogenesis that might trigger certain things. Okay.
Yeah, I think I totally agree with you, Germano, and a lot of research needs to happen in this field. And also, actually, you answered another question which we had from our audience, that two individuals with very similar lifestyle, why do they age differently? So I think that really answers that. Sudha, would you like to put in your opinion on this?
I think I have more questions than comments.
Oh, questions are welcome.
I'm just wondering, as we talk, if we are seeing any selection pressures for carrying on epigenetic markers beyond a few generations. Is there any kind of external selection that ensures it goes beyond a few generations? I don't know if these are questions we have answers for, but I think it'll be interesting. So, because a lot of these discussions are around individuals wanting to have a healthy life, right? So, which means they're looking for answers on what interventions, right? What should I change? Or I know this is something I already have and I can't change it, but what can I change to make it better? And in that context, I wonder, what are those factors that can be changed and what those that cannot be changed? Laxmi or Germano, I don't know if you can comment on those.
And this is also an audience question, so.
So as a clinician point of view, ma'am, I would always say that, the factors which are in our control, right? How we react to certain things, like under the stress, or it could be the current nutrition, our lifestyle. Of course, environment is something which we cannot do anything about it. But the factors which we have in our control right now are the diet and the nutrition, keeping ourselves, as much as possible, taking care of our physical activities, or maybe a reaction to certain factors, so that again our stress can be controlled. Maybe it could be a meditation, or it could be some kind of breathing mechanisms. These are some of the things I would say that which are in our control, and yes, we can definitely try to improve them. But given the expertise, I think Germano would definitely be the better person to answer your questions.
Yeah.
And
well,
I
yes.
already you yourself also.
I think the field of aging has advanced very much in understanding, for example, the concept of biological clock with epigenetic marks. So as a diagnostic tools, this is quite excellent. As a understanding of the process of why we are aging, this is also quite advanced. And I think the point that you're raising is the most important one, how we can go back.
Yeah, how can we remodel? Yes.
And this, I think, it's still very critical. So if we look at in one cell, I think it was remarkable that in the past years, it's called the Yamanaka factor. So the people were able to take a somatic cell that is very differentiated, let's say aged cells, and they force with only 4 pioneering transcriptional factor to go back to a very young stage. Okay, and this works. It works nicely. It works because those factors also are capable of changing or restructuring the epigenome. Now, what is interesting is that when you do those experiments, it worked for most of the cells, but there are still cells that remain refractory. And so I think it's very important, as you mentioned, that we might always have in a population certain individuals that react different to others. And I think those are the ones that we need to look, those exceptions, and not an average effects that we might see, because in those exceptions we might find the key to understand the logic of all those effects. For example, this has been a strategy to look for individuals like that have an extremely long lifespan or short lifespan, and maybe by looking at those individuals that might be even in a population very similar, having similar nutrition, maybe we can try to understand the process and trying to revert to a certain state or decelerate the aging. But I think it's extremely complex, because there is not only one mechanism. There are not few loci. There are not only, and our body is so complex. We have so many different organs, and the beautiful research have shown that in the same individuals, different organs might have different rate of aging.
Yes.
Absolutely.
And so, like it's becoming extremely complex on one hand, but also interesting that we can go into such precise, view of what happened. And so I think, there is still some work to do to resolve these issues, but I think we are on the right track by looking at a variety of system, a variety of organism, having large, cohort. This is very important, because many studies have used certain type of cohort, certain type of individual for certain geographical area of the world. And so I think by increasing, those studies, we might have more reliable outcome, I think, in the precision medicine.
I think I agree with you, Germano, and I think you are rightly pointing out we do need a large-scale data from at organ level, at individual level, at cohort level, of different demographics for us to understand what are the things which need to be remodeled, and then that can be taken up. But that brings a big challenge, and I think, Sudha, you are rightly poised to answer this question. We are doing so much of sequencing, genomics, epigenomics. There's so many big data. And of course, people in the field can analyze that, but still huge amount of data. And for an average user, who's not trained in understanding this kind of genomics data, can AI, so this is again an audience question, so can AI play a role to bridge this gap between the research and the clinic and to the consumer? So I'm putting consumer because now we are not talking about patients, we are talking about healthy aging. So what role do you envisage AI can have in bridging this gap?
So AI, I think, has already made inroads in many fields, not just in the field of aging and health. basically with its capability to essentially process large data sets and potentially identify correlations or signatures that is beyond the human mind to process as an individual, right? So we have computational methods, of course, but that's where definitely AI will play a role. And I think the current interest, as we've been discussing, is looking at a multi-omic approach, which is essentially looking at what's in your DNA, what's in your RNA, what's in your metabolites, your proteomics, right? And so, of course, proteomics and metabolomics are the final expressed forms of what you've inherited or what changes have happened. And how do you integrate these data to essentially arrive at something meaningful, like have a magic pill that everybody takes and remains healthy for a few decades? So definitely AI will be the approach or a way of, going through these large data sets and trying to understand the biology. I think because any biomarker comes with an understanding of the biology, and that's when you go to the next step. So the simple answer is yes, of course, AI will be a critical component of our analysis of large data sets.
Okay. Thanks, Sudha. So I think I'll come to you, Laxmi, but with an additional question. So what are the challenges? So we do have a lot of research. We are generating data based on genomics, epigenomics, and then AI is also being actively used, as Sudha mentioned. It has a big scope. But what are the challenges to translate all of this, research and data to something accessible to the consumer as a healthcare tool, which a person can come, do a simple test, and have all the answers without too much of intervention? Are we there yet? I have my doubts, but considering you are already actively working on this, so what are your perspectives?
So yeah, that's a very good question, ma'am. So again, I wanted to add a little bit on to the previous question to which Sudha ma'am was also talking about. AI actually we do use in our own practice also. So we could see that actually AI is helping us to identify the shifts in the methylation, like understanding it much more better, which sometimes our manual intervention could kind of miss out, right? Like maybe because we are not just using only the epigenetics or only the genetics, but we are using overlaying it with the DNA body metrics and even the life history, pathology matters, so many different levels of information. So based on that, then again we provide a solution. So AI is definitely helping us in in terms of having that huge data at multiple levels, filtering it out. So when we are looking at into future, where definitely we are heading into the positive shift, right? So when it comes to the population and when it comes to scaling it, I think definitely AI will definitely play a very, very prominent role at that time. So currently, I would say the challenging we are facing is there is so much of research out there, right? It is so many things. As of now, when it comes to the aging as such, all we talk about is only the biological, like maybe the Horvath clock for that matter. But I do know that there are different types of ages already, like pheno age is there for that matter. Different clock is also there. So or Grimm age, all this. So when we have this more and more science coming to us for the practical use, I think that would be definitely a much more advantage also. And moreover, to convince not just to convince, but I could go with the causation data rather than a correlation to the client, saying that this is what exactly is what it is leading to, and this is the exact point we need to help you to work on so that we can help you reverse that. So that is one challenging we are facing, and because so much research is still pending, and as of now, even costing is also definitely a factor.
Yes.
So costing. So because India, of course, again India, we do have that as a huge barrier as such. So that's those are the two key challenges as of now we are facing. But other than that, but can see the shift happening really gradually, and people becoming proactive and definitely adapting the new technologies and trying to use those. We are able to use all this data, which so many personalities are working on it, the research is going on. So we are definitely happy to use all that information in our patient care model on a day-to-day practice.
That's very nice that we are on the right track, though the challenges and roadblocks remain, but I think we can overcome that together with such platforms where we bring in academics, clinicians, and entrepreneurs together. So I think what I will do is I'll take one last question, and then we'll open the session to the audience if they have some questions to ask live. And this is a loaded question, and I would request all of you to have your viewpoint. I don't know what's the correct answer. So if the audience question is, are we moving towards a future where an annual epigenetic health checkup becomes as routine as blood tests? And a similar question which we had from the audience, if that is the case, at this current point where it's still based a lot on correlation rather than causation, as you pointed out, Laxmi, what will it have, what impact will it have in terms of anxiety, which will be there in people's mind by looking at all these genomic and epigenomic data, which we don't clearly understand as causation completely? So maybe Sudha, you would want to go first, and then, yeah.
So the simple answer is yes. I think to your first part, will this become a routine test? I think it will. Not maybe this year, but maybe a few years down the line. And you must also realize that people who are doing their annual health checkups are not people who will get anxious about the results they see. They're doing it because they want to intervene and they want to change the course of their life if they can. So I'm not sure how much of the anxiety part will be important, because if somebody's choosing to do the test, then they are likely to be aware of results being this way or that way, right? So I think that should be all right, but definitely the tests are coming. And I think what will spur that is our increased understanding of causation rather than correlation. That's when you can give more definitive answers on how you can, chart the course of your next decades of life. So that's coming as well. And I think thanks to contributors like you and Germano, I think that knowledge base is getting richer. So yeah, I think the audience can look forward to a test that is more reliable in a few years to come.
I think so. I agree with you. Germano, what do you think? What are you seeing in Europe? Is this drive towards all these tests?
Well, France is very conservative. For example, we still don't allow to do the DNA test for ancestry reason. The reason is ethical and also, legally consent to companies to hold, our data. So France is a little bit peculiar on this, but yeah, in US, I mean, you go online, you find all these epigenome tests which are based on DNA methylation, and was developed first by Steve Howard, where they identified these 353 loci methylated. Now we have more. I mean, it's becoming more and more accurate. I think as a predictive tool, there is nothing wrong. I think it's good to know, what will be our, fate. Maybe there are some global intervention that might work or not. So, but certainly they don't harm you. So I still think is positive. Testing is positive. Being aware is certainly positive. But of course, how much knowing that these loci are marker of something, how much can be causative, this is difficult. The good news is that I think with the CRISPR technology and epigenetic reprogramming, where we can reprogram individual loci, now the research has been going, really fast in understanding whether such specific marker that we use for biomarker, they might have a functional role. And I think in this perspective, maybe, in a few years, we will also know which one among those markers that people are testing might be really functional, might have an important things, and whether we can, revert or taking some specific action in each individuals, right?
I agree. And I agree that we need to have good ethical guidelines in place when we are working with such big data. Especially, we don't want insurance companies to start using these data to hike up the premiums. Yeah. So, Laxmi, the last word on this topic from you, and then I will probably take a few questions from the audience. We are almost out of time, but maybe a couple of questions we'll take. Please, Laxmi, go ahead.
I just add on to the point. So if you look at our population, Indian population, 15 years ago, right, nobody would even go for a blood test unless it was needed. Right? People would not say, I'm completely fine, perfectly fine. They will not do it. But right now, that is not the scenario. Whether they, everybody is more in that proactive nature and getting those annual checkups, and also actually going for those just for the sake of it, just to keep that everything is under control. So they are going for those routine checkups. So awareness has improved. The shift has already started happening. So now that awareness about DNA and the epigenetics, I can see that happening in my course of last 10 years itself. I can see the shift happening. People are now actually accepting and very, very proactively coming to understand what can they do more. I'm not sure if it is because of the post-COVID, but people want to be ready. They want to know what could they do a little bit extra to keep themselves much more healthier, right? To just create that healthier version of that their selves, right? If it is at their fingertips, why not? So definitely I can see in the future people doing the epigenetics just like any other blood test for that matter.
Right, absolutely right. So what I will do is we have some questions in the chat. Maybe we can take some of them. Sagarika, is it possible that the audience can unmute themselves and directly ask, or I should read the question?
No, ma'am, it's better you read the question because in the interest of time,
yes,
that won't be possible.
Okay, so I think one question which we have, and I think many of these points we have already covered. So especially for you, Laxmi, there is a question that what is the most easily measurable and trackable epigenetic mark which can be utilized? So basically, in practice, what you are utilizing right now to modify the lifestyle changes and exercises.
Right. So so in my career, like at our clinic, we actually tend to definitely both to the athletes and non-athletes as per se. So when it comes to the non-athletes, I'm definitely looking at a wellness point of view and helping them to better maintain their disease condition. And again, age poses a very, very important factor in that, right? So the one measurable we could see is definitely how the aging has aggravated because of the disease condition. So we could track that, and post intervention, like it could be the sleep, the diet, and the supplementation, we could measurably show that how we could reverse that age in terms of 3 to 5 years. When it comes to the athlete point of view, we can see the performance for them is again age day to day, right? So again, with aging, definitely the energy levels will be slow. So then that performance takes a toll. So again, with the epigenetic and the intervention tools which we give them post the assessment, we could significantly show them the improvement in their performance and also the reduction in the injury likeliness of any injuries happening.
Thank you, Laxmi. So another question which we have, and I think we already had a good discussion on this, is that when we're talking about transgenerational inheritance or even intergenerational inheritance for that matter, so the question is, are we only talking about beneficial modifications and humans or organisms can filter out the detrimental ones, or we inherit both? What is your take on this, Germano?
Can you repeat the question? Sorry, Meetali.
So basically, when we talk about inheritance, right, in response to stress in the previous generation, is it only the beneficial one, or we're also inheriting detrimental effects?
Yeah, I mean, yeah, thanks for asking the question, which is a very debated question. So we tend to to agree that, there are certainly, some detrimental effects that might inherit from our parents. And this was the case of, few metabolic disorder I mentioned, the impact of the microbiota. Those can have, consequences in the next generation, negative consequences, stress, all these study in mice, stressing the parents through the sperm, transferring something that might adversely affect the next generation. But we also have positive effects. Many studies, including our lab, we see that, like the stress resilience phenotype coming from parents adapting to certain type of stress can be heritable. So there are certain components that might have beneficial and adverse effect. And that's why, it's difficult to disentangle what can be good, what can be bad. We cannot globally intervene on a mechanism that let you inherit both good and bad, because otherwise we block what can be potentially bad, but we also block some other type of inheritance that might be very important. And so this is why we need to go into understanding the specificity of those type of heritability, which genes we are talking about, which molecules and where do they act, more than, globally affecting those pathways that might certainly interfere with the process, but also block some other very important heritability process.
Yeah, I think very well said, Germano. And I think I have one last question for you, Sudha, before we wrap up today's session, because I also don't want to take all of your time. So the in and out of genomics field in India. So the question here is, how much of large cohort studies are happening in field of epigenetics in India? Are we still relying a lot on Western data?
Yes, we infancy. We don't have large cohort data yet on epigenetics. We have done some recent studies, for example, on a cohort diabetes, where they had a longitudinal study of 6 years. But that's again not a very large data set, but nevertheless very interesting insights even from that small cohort that we have. There have been some studies on aging with respect to neurological disorders, dementia. Again, these are all studies that are ongoing. We don't have data in the public domain that we can use for, developing like what Laxmi is doing. We're not there yet. It will take us some more years to get there.
Okay. Well, I think we have a good start, and now we have to pick up the pace.
Absolutely.
Yes. So I think in interest of time, there are a lot of questions in the chat, and maybe you can go over it, and audience can continue the discussion later on. So I will wrap up this session today, and I must say that I got to learn a lot today from all of you. We learned about basic definitions of epigenetic mechanisms, how our lifestyle can impact the epigenetic, where we are in terms of the technology which is ready to use right now, what are the challenges, roadblocks, progresses which we are making. So a wonderful session, and a lot of questions from our audience which made this session very fruitful. So thank you to everyone attending, and thank you especially to you, Sudha, Laxmi, and Germano, to take out this time from your very busy schedules. I really appreciate it. Thank you so much for joining us today. I request all the audience to give a big round of applause. You can use that clap emoji option in your Teams to show the appreciation to our panelists. And thank you, Sagarika, for managing the webinar very well. Thank you so much. I look forward to seeing you all again.
Thank you, Meetali. Thank you, Sagarika.
Thank you all the panelists.
Thank you, everyone.
And thank you, Meetali, ma'am.
Most welcome. Thank
It
you.
was very interesting.
Yeah.
Thank you. It was great listening to both Sudha ma'am and Germano. Very great moderating, Meetali, for sure. The compilation of all the questions, it was truly, I would say, definitely quite a lot of insights there.
Yeah, I give the credit to our audience for these insightful questions, and Sagarika to put them together.
Thank you.
Thank you.
Thank you so much. See you then. Bye.
Bye-bye.